Very much. Good afternoon, everyone. Very, very nice to be here. Nice to see you all. My name is Videha. I'm a doctor and innovation lead at the University of Manchester. I'm going to be talking to you about continuing the theme around patient-centered care and about thinking about patients as individuals and not just as pieces of steak that have got no flavour. We have been working on using genomics to try to personalise treatments. Now that sounds quite futuristic. It's been very, very new to me. It's something I didn't know a lot about myself. I'm actually a surgeon by training, so I'm familiar with steak. But thinking about this has actually been super exciting for me, and I've become increasingly passionate about this because you can see the opportunity for improving care and outcomes by being more personalised, not just when it comes to decision making, but also when it comes to treatment and actually influencing how individual people react to different medications. So this is going to be a double act. I'm going to give a bit of introduction from the clinical and data requirements perspective, and then Ian is going to talk about some of the work that we've been doing to try to support solutions that can actually enable precision medicine to be implemented in routine clinical care. Great, so I'll give you a little bit of background. As I said, this is a topic that's relatively new to me as well, so hopefully we can learn together, and then talk a little bit about the requirements both from a clinical perspective as well as a data perspective, and Ian will take us through some of the work that we've been doing to actually start to build some data models to support implementation. So, as you will all know, we all respond differently to medicines. Sometimes medicines work well, sometimes they don't work as expected, and sometimes we have side effects. We know that changes in our genetic code and our DNA play a role in influencing that response to medicines. We've known this for a very long time. Science in this space is well over 30, 40 years old, and we know lots of individual gene-medicine or gene-drug relationships that are established in the scientific literature. And this relationship between our DNA and our response to medicines is known as pharmacogenomics. Pharmaco coming from medicines and genomics coming from genes. Sometimes abbreviated as PGX, that's people in the pharmacogenomics world often talk about PGX. But in plain English terms, it's personalised prescribing. It's prescribing medications based on individual characteristics and individual traits. If we were to test for these gene changes, we would be able to personalise prescribing and therefore make medicines more safe and effective. Now, when I learned about all of this, I was obviously very curious. Oh gosh, this sounds great. But at the same time, I thought genetics, this is about rare genes or things that are not very common. This is not really relatable to everyday healthcare. And then when I started to learn more about it, I actually started to realise that this is extremely common. So all of us in this room will carry over 99% of us. This is based on very large biobank studies. Over 99% of people carry a genetic variation that influences how they respond to medicine, which is an interesting concept because that means that this is not a genetic abnormality, and people often think about a difference in individuals. Gosh, you don't respond to this medicine well, you are not normal. And even some of the language that is used within the pharmacogenomics world, people talk about normal metabolizers and rapid metabolizers or slow metabolizers of certain medications. I think that's actually quite right. This is common variation that is just describes how we as an individual respond to medication. If you are a rapid metabolizer, it doesn't mean that you're unwell. It doesn't mean that you've got a disease or a condition. That is just how you break down a particular medication, how you break down a particular drug that you take. And I think that's important because, and we'll come to that when we start to talk about this from a data perspective, this is maybe a slightly different concept to lots of other medical information we store in our electronic health record. This is not exactly a diagnosis. It's not an illness that you have. It's maybe not exactly an observation either. So exactly where does this fit in within our existing constructs of how we describe medical information? So despite knowing all of this and it all being very exciting, we don't do this in the real world. Medicine is not personalised today. Any of us that goes to see a doctor, the doctor will say, well, gosh, for you, here's you've got high cholesterol, here's a statin, or, gosh, you've been feeling anxious, try this antidepressant, and hope that it works. Sometimes people describe that as trial and error, or giving things a go and then seeing what happens. But we can do better. We know that the prescribing of medicines is the most common therapeutic intervention in healthcare. It's something that I, as a doctor, is the most common thing that I do for patients. If it's an active thing that I do, which is to prescribe a medication. And then, as I said, this is about common changes in genes that we all carry, and this is about common medications that we are prescribing regularly, and lots of us take. And I'm sure I will take some of these medications during the course of my life. Painkillers, antidepressants, blood thinners, statins, medicines that are prescribed at scale. So if we then think about this, we've talked about the individual patient and the individual benefits and care and outcomes for an individual patient. If you then think about it from a healthcare practitioner's perspective, and we spent a lot of work talking to healthcare professionals about this because, as you guys will be very familiar with, in kind of a big part of the open-air philosophy, a big part of the open-air culture is to be user-led, to be led by the needs of the clinical service or of patients. So we spend lots of time with GPs, pharmacists, out in the community trying to understand how this relates to their everyday practice. And one of the things that recurrently comes back is that healthcare professionals say, gosh, I give my patient a medication and they go away, and then 6 weeks later they come and see me again to say, gosh, doc, it hasn't worked, or can I see your pharmacist? They can maybe change something, or alter a dose, or try something else. And that is an additional healthcare burden. That's an additional appointment, that's additional time that a healthcare professional has to take. It's another consultation that's taken away from someone else who may be more in need, certainly if that is something that we could potentially avoid. So I feel like this idea of using genomics to personalize medication, particularly in that primary care, the place where medication is prescribed at scale, where common medications are prescribed, is a real health opportunity that we haven't really achieved yet. And it aligns with some of the other general shifts in healthcare, moving towards more preventative models of care, trying to be more community-based, and indeed this expectation from patients. They want to have more choice, they want to have more control, and patients want to have more confidence that the treatment they're getting is tailored to them. That idea of being more consumer-oriented. All other industries are empowering consumers, are putting individuals much more in the driving seat, and we should be doing that as in healthcare as well. And this is just to share that this is being translated into policy, and this is being translated into recommendations. So this is a recent guideline that was published by the National Institute of Health and Care Excellence, or NICE, which some of you will be familiar with, which is the main guideline producing body in the United Kingdom. And they've now recommended, just earlier this year, that every patient that goes to hospital with a stroke or a transient ischemic attack, a mini stroke basically, a period of time where you lost function but then you recovered, should have a genetic test to guide the use of clopidogrel, one of the most commonly prescribed antiplatelet drugs. And this is exciting for me because I find this space interesting, but it's also exciting from a genetics perspective because, as I said earlier, genetics typically been used in rare diseases or in cancer. But this is genetics, the clinical front line. This is genetics in A&E. This is genetics on a ward, which is really different to how we've traditionally or historically thought about genetics. And that takes us to some of these clinical requirements. Because there is a shift in where this genetics is going to be used, there's a shift in the people that are going to be delivering that care. So the people that will be using this type of information are not cancer specialists. They're not geneticists. They're not children's doctors who work at a tertiary centre. This could be your community doctor who works out in a GP practice. It could be someone who works in an emergency department. It could be not a doctor at all. It doesn't have to be a doctor that is perhaps interpreting this information. So the end users are completely different to how the end users that have previously used genetics. And therefore, we need to think about this in a completely different way. Traditionally, genetic labs, and I talked to someone about this earlier, the typical output from a genetics lab's perspective is to produce a report. We produce genetic reports. That's the most typical thing. But a report is a long, 2, 3, 4 page document with lots of information, lots of detail, lots of nuance. An average GP or a doctor working in the emergency department has not got time, knowledge, or experience to be interpreting some kind of genetic report. So one of the things that we've been talking about, and this is a big culture shift, not just for the prescribers, but also for the labs, is to say, I know you're used to producing reports, we want you to move to producing results. which are short, discrete, structured, and standardized. And that is a real big mindset shift from oh, here's a 4-page PDF that we'll email to you, to, oh, now we need to think about structuring this data and organizing it so it can be placed into an electronic health record or power clinical decision support systems. So yeah, so that feeds into that last comment there. So clinicians only really want to know the clinically relevant information. They don't need to know all the detail and all the history and all the lab stuff. And that's what we found when we were going through the data modeling process. You have these different perspectives. You have people from the laboratory world who would not understand that, who wouldn't get that, gosh, this is not clinically relevant, it's not clinically important, it doesn't make a difference to the decision-making process. So even though it might be derived from raw genetic data, it is distinctly different. The pieces that drive clinical decision-making are different. And of course, as we already heard, healthcare professionals do not want new portals and web pages and logins, and we all know this, right? So whatever we do needs to be presented within existing systems and part of existing workflow. So that is, that is an absolute fundamental part of any kind of implementation of innovation in healthcare. And there obviously comes lots of the challenges around integration and legacy, etc. So from a data perspective then, and this is my last slide before I hand over to Ian, another few interesting perspectives for me were the fact that, well, gosh, genomic data is for life. It's slightly different to other healthcare data that might be relevant or useful for a particular healthcare episode. This is data that's useful for your entire lifespan. Also, your genes don't change over time, right? So if you, and certainly the genes that relate to how you respond to medicines don't change. So if you have had a pharmacogenetic test at some point in your lifetime, that should inform prescribing throughout. So being able to store that in a longitudinal way, in a structured way, is really, really important. The sequencing machines that do this testing produce a range of different outputs, and they're not standardized, but we are in a fortunate position. There are not that many different companies that produce these machines. There's probably only 3 companies that produce genomics, and that might change in the future, but certainly for now, that is a manageable number in terms of building automations or building solutions that are able to standardize outputs from these genomic sequences. It's not like we've got 50 machines or companies that are in this space, good or bad. And that is because there are no standards in that space at the moment. If there were standards in that space, of course, that'd be different. You could have more innovation, you could have more competition, but at the moment, we don't. And of course, we know that the health IT landscapes everywhere are varied, and within the NHS, the context where we are working, that is of course also the case. And similarly, lots of electronic health records that are implemented will have built-in or have add-on clinical decision support modules. So for example, here within the UK, within primary care, we have it's a duopoly of 2 main IT suppliers that provide the electronic health records, and then there's probably 2 separate clinical decision support companies that integrate with each of those. So we know that there is going to be a need for this information to be used across different care settings, different disparate range of end users, new end users, and that is in the context of lots of different health IT integrations. So we said from the outset, we need a vendor-neutral, independent, agnostic platform approach in order to operationalize this. And this was met with, as you can imagine, quite a lot of resistance, particularly the primary care IT suppliers that we worked with. They said, "Oh, don't worry about it. Just send us the data. It's fine. We'll build the clinical decision support. We'll put it in. We'll create SNOMED codes for you. Don't worry about it." And we were like, "No, thank you. That is not the approach that we want to take. We want to be, independent. We want to be interoperable by design. We want to keep the data ourselves, and we'll expose it to you as an API." And this is just me kind of doodling and kind of drawing of what I kind of came up with, of what that kind of would look like from a kind of schematic perspective. You've got your genomic data that's produced in a lab. We then have a short automated bioinformatics pipeline that translates that raw genomic data into clinically actionable results. That is then linked with a knowledge base that interprets these individual results, which go into a live database, and then we expose that as an API. And we use something called CDS hooks which is again an HL7 standard to drive clinical decision support systems. So that's, again, this is just me drawing, right? So just me messing around. But that's how we kind of conceptually came up with what that should look like. Okay, I'm going to stop there. I'm going to hand over to Ian for the more technical and hopefully interesting part. Thank you.
Not that technical. Steady yourselves. Yeah. So at this point, this is this is where we came in as FreshEHR. Most of you know me, but we do openEHR, archetyping, templating, etc. And the question is, if we get a message on the left, something like that from a lab, and as Videha said, they are different. There are no standards here. What do we do? How do we turn that into an archetype? What does it look like? Do we need to capture all that information on a patient record, and where should it go? But it is very lab test-like, and the original intention was always that we should treat this as a lab test primarily, an extension on the fairly mature lab test mechanism that we have in openEHR, which itself is based on HL7V2 and whatever. So we're not doing anything radically different there. But we got this pipeline of the pharmacogenetics all the way through that leads to something like that. As you can see, there are some bits that won't make any sense, but fundamentally we've got the gene name, we've got a genotype, which made little sense to me, and I still struggle with some of these things, to be honest. We've got the phenotype, and that's the critical bit of information. That's the thing that drives everything else. What you'll notice is there is no drug name in there. And this was a difference that we noticed between particularly the US way of thinking and, from what we can gather, the EU or European way of thinking, is that they tend to test against the drug. So in the example that Videha talked about, they will say, what about clopidogrel or codeine as a drug, or maybe a panel of drugs, whereas the lab practice here is much more about testing against a gene or a panel of genes. So you get these cultural differences as well. So that was our job. Develop archetypes, we didn't know quite what, and or templates. Definitely aligned with the standard openEHR lab pattern. We want to be in that or thereabout. We wanted to, there has already been work done previously by a lot of people, particularly from Sardinia. Giovanni's sitting in the front, and others, the HiMed group as well, Norwegian colleagues as well, around openEHR CKM genomics. We'll come back to that in a little bit. We're also aware of work that was done by the FAR community, and really nice. This is one of their diagrams, very pretty and nicely labelled, although I found a bug in their labelling. But one thing is quite interesting. I mean, those of you familiar with FAR, it's all quite spread out. It turns out into 1 million different profiles, and it was very much about how a lab would send the data from the lab to the consumer. It wasn't about how you would actually store that in your record as the lab test comes in. So we want to be the target of this thing, except it's likely to be coming in with a FAR or FAR-like message in the future. So this coloured our thought, and certainly it made a lot of the challenges clear. And we also wanted to make sure that we could get the data out again in that format, because NHS England, they want to use a variation on that. We also wanted to take this through CKM publication. We originally thought about doing this in the UK, and then we thought, but that's crazy, If we get this right, this really has, this is absolutely international. But we needed to engage the PGX community. We needed to engage a whole lot of different people. So that was our challenge. And the main thing was, what should actually be part of the clinical record? What's fluff? As Videha said, there's a lot of detailed information. There's, sometimes you get a whole sequence. Sometimes you get a list of variants, which mean absolutely nothing to me. Then you get what significant genes do I have, so that's the genotype, and that starts to make a little bit more information to a clinician who's maybe used to that. Then you get down to the payload, the phenotype, the metabolizer, and that's the thing that's lifelong. Then, of course, out of that, you get derived what the FAR guys call therapeutic implication. Of course, that's the decision support. But we decided that we wouldn't go to the decision support layer, because that's derived and it will change over time. The rules, the understanding, new drugs will come in there. That's the thing in the middle, that your metabolizer status on that drug that drives it. And much of this work has already been done by several groups, I think, but the main one is in the US called CPIC. So they have all the rules that say, if you've got that, that becomes this metabolizer status, and these are all LOIC or SNOMED coded. And if you've got that, then these are the drugs. So they've got spreadsheets and panels and databases. So this is all real. You know, we can do this. It's just, what do we do with the data? Well, we get it from one of these systems. So this is what we came up with, the idea of a pharmacogenetics test result cluster. And I won't go through the details, but it's basically what was on the screen above. We will capture in a little bit more detail, but this is designed to sit inside the normal lab pattern. Those of you who know there's a lab test and then there's an analyte. This sits as underneath that as a kind of extension on the lab pattern because it has these very specific little fields, the genotype, the metabolizer status. Of course, the GP systems were lying. They can't handle this because they can't handle this kind of complex data. You know, they can handle a code and a date and maybe a value, but anything else becomes a mush. So they can't actually do it unless you overload this to SNOMED, which is a bad way of doing it. So, and ultimately this will turn into some kind of pharmacogenetic diagnostic report. That's the template. But how are we going to get this through CKM? Now, you all know, you know this world. CKM editorial work is fantastic. But we know that if we rocked up and said, hey guys, hey Bjorn, who's our new CK lead, hey, we want to do this thing, how long will it take? It would go into a schedule. Quite rightly, there are many other priorities. But we had money, literally, we were, as FreshEHR, we were given a little bit of money, a small bit of money from Videha's group. And we said, but we actually have the editorial skills. We're not the current editors, but we've done this before. And we thought, why don't we suggest a kind of subcontracting arrangement that we'll try it as an experiment. Right? If you've got a good use case, a group of responsible editors who know how this thing works, why might we experiment with the idea of subcontracting some of that CKM editorial work out to a group who've got, are recognised as having at least the skills to give it a go. So my colleague Heidi Koikalainen from FreshEHR, she led the CKM work, but with a lot of input from CKA leads like Bjorn and Celia joined the calls as well, kind of as reviewers. So we regarded them as not as editors because we didn't want to land them with a lot of work, but they came and sat in on the process and the understanding. But what was fantastic, and I think it's an absolute joy to see this process working, is we brought together the openEHR modelling experts, which is us, people like Videha and John McDermott, who are world-leading PGX experts, people like Giovanni and Sheila from Sardinia, but other frontline clinicians. And for all of us, including the genomics experts, this was all pretty new because it's applied genomics, it's not the raw research genomics. So there was a lot of conversation, there was a lot of misunderstandings, but we got there, we got around it. And it was real proof that this is the only way you can just wrangle these discussions in a really small use case, and we got to the end. So we're about to publish this archetype, and again, we wanted to do this and give the CKA leads some reviews. So a little delay in publication because they came back and said, no, we're not entirely happy about that. So it was always recognised that CKM International was in control, or the team there were in control, but we were working under their licence, if you like. I think it's like, when breweries brew Carlsberg locally, we're the local brewery, but the Carlsberg guys are keeping the recipe right. I think it's a model that we could actually think about using more, particularly as more people get these kind of editorial skills. But here's an interesting question. So Videha said, of course, this data is for life. Your genomic test is for life. It's actually not. And this is what we suddenly realized in the middle of this thing, because these are targeted tests. This is not like a whole sequence of DNA that they lift out and they analyze and say, they actually target and they look for variants. And not every test looks at all the variants. Broadly speaking, the good tests do all the variants, but if you go to a local pharmacist and say, can you take it, they may have a cheap and cheerful test that only looks for the two common ones. So the reality is that you may end up with having multiple tests over time, and what you get is a patchwork. You'll get the common ones every time, but you may get some of these other variants that are significant, less common, but being picked up over time. So we've not got actually one source of truth, we've got a patchwork of tests coming in. So what we now think is we probably need something around an evaluation which allows us to aggregate that together. And Videha's already said, the question is what should that be in openEHR terms? Is it a problem? I think so. Is it an allergy? Definitely not in my view. Is it a precaution? Maybe. But we tend to think of precautions as abnormalities or problems, whereas Videha said, this is sometimes just a statement of normality. This is how EPIC have solved the problem. And this is the only system I've seen that have actually addressed it. They have an idea of a genomic indicator, which is a new part of their record. And yeah, Bjorn's looking very concerned. But we are going to start this conversation basically to say you can't read it deliberately. You know, so you can, I don't want to upset you. I think we probably need a new archetype called a genomic indicator, one flavour of which might be a PGX indicator. But it's just fascinating that where this work takes you. You know, maybe this is my first go. It will actually carry that pharmacogenetic testing data inside it, but it has a kind of headline, you know, the overall metabolizer status and gene status, and probably some bit of text in it. And this will be the thing that triggers decision support, not those individual tests. And once we started reading the research, there's quite a lot of good research in terms of EHRs. People have been down the same road, including the people working with EPIC. So we suddenly, well, oh my God, we're not the only crazy people that are thinking in this. Okay,
Perfect.
so that's it.
Thanks for David. Thanks, Ian. Right, we're going to round this out with some questions, some really hard killing questions. Right? For Bjorn, maybe you would like the opportunity to respond.
The reason I looked skeptical was I couldn't read it. That's an interesting idea. We'll have to discuss.
There'll be a long discourse conversation on this. And then we said that to our pharmacists. We said, look, guys, this is not your problem. This is ours. You know, this is now we're into an informatic conversation. It doesn't. The genomics doesn't influence this. This is our problem to resolve. It's an interesting. It is. I think it's Videha said. I gave up GP practice 20 years ago, and as we got into this a little bit, I suddenly got excited about clinical medicine. You know, because an awful lot of the stuff that gets thrown, you get the whizzy stuff, you get the AI, you get the genomics, ultra precision, and I thought, yeah, it's too hard. It's going to make my ears bleed. But as we started to understand how it was delivered and what is delivered, it's really high value information. It's saying, don't give that drug. There's no point. It won't work. 50% of people do not respond at all to codeine. And we can tell you, that's why we don't give them codeine, it's pointless. Give them clopidogrel, but give them half the dose, otherwise you'll poison them. So it's not like even allergies stuff is a nightmare, because it's all very nuanced and makes you have to think. This is quite easy to process and act on.
I have
Trouble.
in children, you test for G6PD, glucose 6-phosphate dehydrogenase.
Yeah.
Would you include that in that same? Because that is considered a diagnosis today.
Yeah.
But it's actually a genetic testing that says don't use these drugs because they will have a lot of this.
Do you want to take that?
Yeah, sure. I mean, look, I think a lot of these things are so G6PD is obviously really well established. It's historic, and it's the name itself is the D stands for disorder. So it's it's seen as a abnormality or a diagnosis, and I think as time has gone on and we've kind of evolved and changed, and I think this is very new. The fact that we're saying this is not an abnormality, no one said that before. No one's thought about these concepts in that way, and this is why we are keen to present it, because we don't know what the right answer is yet either. This is really emerging. This is the kind of bleeding edge of kind of healthcare innovation, of trying to figure out how do you do this in the real world, not for things that have historically been done. How do you do this for new stuff? And how do you make it happen on the clinical frontline where prescribing happens? So yeah, I mean, it's very, very good. Maybe we should look at G6PD to see what we can learn from how that was done. But yeah, I'm not sure exactly in terms of how that relates to what we're trying to do with all these other common pharmacogenes.
And the EPIC genomics indicators thing is actually much broader than, so they'll be using it for things like G6PD or DYPD or some of the significant cancer. They said we need to just highlight this. This needs its own space in the record. Yeah. I'm not saying that's right. I'm not sure myself, but I think that's what we need to consider.
And it's been very interesting because most of the mature implementations are in the US. So we did like a large international review of all the pharmacogenetic implementations in the world, and over 90% of pharmacogenetics happens in the US. But of course, they're all institution specific. right? So there's in one hospital where there is one electronic health record, the lab is within the same building, and they are doing, in workflow, clinical decision support, patients are having, personalized precision treatment. But, that is the flagship advertising for that hospital. We in Europe don't work like that. That's not how we deliver healthcare. It's not like we say, one of the kind of jokes I always make it in the NHS, like in the US, people are like, come to this hospital, we'll advertise, you have precision medicine here, it's really special. In the NHS, we're like, don't come here, go to that hospital, we're really busy. Like, we've got beds, like, go away. So it's like completely the opposite. And we want to deliver equitable care, right? We don't want to have, here is really special, if you go here, you'll get the best care in the world, and if you go there, you die. Like, that's not how we want to deliver or design healthcare, right? So we have to think about things really differently.
Well, I think that's a great point to end on. And on behalf of the whole team, I'd just like to thank you for your participation, but also our presenters for all their hard work. And I'd just love to thank Abby as well. I think we should all thank Abby. She's done an amazing job.